Hi, this is Margaret from the CBM. This video is going to focus on the biology of drug resistance. When we talk about resistance, we're actually talking about a change in the organism to which the drug is targeted. So it might be a virus, it might be a bacterium, it might be a cancer cell, it could even be a weed that you're trying to get rid of with a pesticide. So the short answer of what causes resistance is evolution. Typically, what you're trying to control is a large population. There's variation within the population. This is due to differences in the genes, which is due to error-prone repair. We'll talk about this a little bit more in depth. Because there's a large population, there's competition for limited resources, and only those that are the best adapted to that particular environment will be able to survive long enough to reproduce and pass their traits on to the next generation. So this is a very brief overview of how evolution of a species can lead to resistance. You continue to expose the species to the drug, and those that have variation in them that make them less sensitive to the drug will survive and reproduce. Okay, but let's go in a little more deeply. So first of all, you get variation because when nucleic acids are replicated, there is often an error-prone replication process. In particular, you get this with viruses that have error-prone replication machinery and bacteria. Viruses in particular don't have, a lot of them aren't double-stranded nucleic acids, and therefore there's not the complementary strand to correct any errors that are made. So this results in a lot of variation within a species, within a population. The variation is in the DNA or the RNA. Some of the viruses are RNA viruses, and therefore I'm saying nucleic acids more broadly. And within this population, there's a large variation because of this error-prone repair. Then when this population is exposed to a drug, the drug prevents the function of proteins that are susceptible. So it binds to the proteins that it binds to, and those organisms die. But those organisms that have a variation in the protein from the variation in the DNA or RNA they don't bind the drug, and therefore they survive to reproduce. Then when you're exposed again and again to that same drug, when the organism is, you're continually selecting for those organisms that are most resistant to the drug. And so over time you get drug resistance. There's a number of factors that contribute to the development of resistance. The first one is that the cells or the organisms that we target have a faster generation time than we do. Once a virus gets in your body, it replicates very quickly. It can sometimes produce, one virus can produce, say 200 viruses within a matter of an hour. And then each of those infects another cell, and in another hour you have 200 times 200, which is 40,000 viruses. So if each one of these is different or has some slight variation, some of them may be resistant to whatever you're using to treat the virus. Bacteria also have very rapid generation time. In good conditions, a lot of bacteria can replicate every 15 minutes. And if you're trying to design a drug to fight cancer cells, the cancer cells within our body typically one of the reasons that they're cancer cells is that their replication has gone out of control, and they can replicate every 24 hours or so. Whereas most of your cells in your body, if you're an adult, don't regenerate that quickly. Air-prone repair is another big issue, particularly with viruses. As I mentioned earlier, viruses oftentimes are single-stranded nucleic acids, so they don't have the complementary strand of DNA to use as a template. If there's a mistake, they don't catch the mistake. Some of the enzymes that are involved in replicating DNA are also air-prone. And viruses, for instance, within an individual become what we call a quasi-species. So if you look at someone, for instance, with HIV, this is a retrovirus that gets inserted into the cells, and as it replicates throughout the body, it exists in the body over many years. And if you look at the HIV in one individual, there are a number of different variants of that virus. And you can actually track where someone got HIV based on the presence of variations within the genome of the virus. Bacteria are notoriously promiscuous. And what I mean by that is that if you consider, say, the biology in your gut, the bacteria in your gut, you have a very, very high concentration of a wide variety of organisms all living together in your gut community. And bacteria are very good at being able to scavenge DNA from other cells from the environment and swap DNA among species. So for instance, through transduction, a virus can carry DNA from one bacterium to another. And then through conjugation, bacteria can swap DNA with others. And even through something called transformation, bacteria are able to pick up naked DNA from the environment. So let's assume, for instance, in your gut, you have 500 species of bacteria. And one of those bacteria has come up with a variation that is resistant to a drug. And you're treated with that drug. And that particular bacterium will survive and replicate better than not only other bacteria within the species, but within other species. But then over time, your body gets repopulated with the bacteria that were wiped out when you took the antibiotic. And the one bacterium that was in a better position to survive because it had this resistance gene, maybe in the new environment without the antibiotic, it's not as able to reproduce, and the bacterium dies. And all of its progeny dies. So instead of having one bacterium, you have thousands of bacteria that die because for some reason, without the antibiotic, they're at a disadvantage. Well, when those cells break open, their DNA is spewed forth in your gut. And other bacteria then are able to, through transformation, just pick up that DNA. And it might get incorporated into their genome, and it might also carry that variant that makes it resistant to the antibiotic. So later on, you get sick again, you take the antibiotic again, and now it's just not one bacterium that's resistant, but many different species within your gut that are resistant. They survive, replicate, and it goes on and on. So once one species of bacterium develops resistance to a drug, because of this promiscuous swapping of DNA across species, very quickly, most bacteria carry those resistance genes. And often there are cassettes of genes that render resistance to many different drugs. And so we keep building up these more and more resistant strains of bacteria that are resistant to a whole lot of different drugs. So this concludes the video on how organisms develop resistance. And the third video is going to explore a better way of designing drugs to reduce the likelihood of resistance, which is Dr. Schiffer's work. I'll see you in the next video.